Hi, this is Miss Litton, and this is a very tiny little crowd at lunch. Say hi. Hi. And we are reviewing this is the APBio unit.
Hi there. Just in case you didn't hear, attention all students and staff, please join OPHS
Just a moment.
counselors in G9 today.
So anyway, sorry about that, but this is the part 1 review for unit 2 essays. So the first thing I want to bring up is the PDF. And remember, these are not organized per our units. It's just a list of potential questions, like content-based questions. So our unit starts with the membranes, right? Okay. So this first one says, describe the model of a cell membrane of a eukaryotic cell and discuss different ways in which substances move across the membrane. In your discussion, be sure to indicate whether your examples are passive or active transport. So for that type of question there, and in fact, I'm gonna grab all of these real quick for you, so we can break them down. So I'm going to go into a new notebook here and paste that. Could you, while I'm doing that, could you think of how would you diagram out a cell membrane? What kind of things would be critical for you to have in a cell membrane? Cell membrane. Think about a cell membrane. What would you want to put in your cell membrane? Talk it over or put it in your notes that you have right now. Are you sharing? Or you have some notes? Okay, so I'm handling this first one, and so if I was going to draw a cell membrane, okay, I would probably have some of these in it. What are these? What? Phospholipids. And then I might have something like this in it. What's that? Yeah. And what would this be? Peripheral protein. I might have something like this in it. Cholesterol. Yeah. But that would tell you something about this particular membrane, that it was what? What's the only types of cells that have cholesterol in their cell membranes? Animal cells. Okay. I would also have some stuff coming off of these potentially. What would that be? Glycolipid. Yes. Yeah. This would be a glycoprotein, and this one would be a glycolipid, part of the glycocalyx or the carbohydrate side chains. And then I might also have some of this here. What would that be? Cytoskeleton. Good job. So things that I would also probably want to identify is I want to say that this area right here was one thing, and this area right here would be considered something else. Yep, exactly. I would say this region right here is nonpolar, and these regions right here are polar. Okay, do you know where the inside of the cell and the outside of the cell is? Okay, so what are the 3 ways something would cross our cell membrane? If I go like this, let me see. There we go. Okay, what are the 3 ways that something could cross this cell membrane? Do you remember? Tell me.
Through the membrane.
Yeah, so you could go through the phospholipid bilayer, right? And then when I'm talking about through the phospholipid bilayer, the only ones that would do that is if they were what? Small, and what else? No charge. They can't have any kind of charge, otherwise it couldn't go through the phospholipid bilayer. Okay? Now, what do we call it when if it's going to go through the phospholipid bilayer, then what process is it doing? Starts with a D. Diffusion. Okay, so this would all be diffusion. Okay, if it's water that's doing the diffusion, what would we call it? Osmosis. And when we're talking about osmosis and water, then this is when we're talking about water potential, right? And we're talking about hyper, hypo, right? Iso. Those words should all come to mind when we're thinking about water going across it. And what is a water potential equal to? Pressure potential plus what? Solute potential. And pressure potential, then we're usually involving cell walls. And when I think about solute potential, then that makes me think, yep, all right. So those all words, those come into play when you're talking about diffusion, going from a higher concentration to a lower concentration. So this would be, you know, one way to go through it is through the phospholipid bilayer. Okay, what would be a second way to go across it? Yeah, you would go through some sort of protein. Okay, so this would be our number 2, going between the phospholipid. That would be our number 1, right? So using some sort of protein. Now, when we use a protein to go through, we're either talking about, and I heard you say it, what was it? A channel or a what? Carrier. Okay? Now, channel or carrier, just does it bind to it or does it allow it to go through, right? And so when you're using a protein, and if we're using a protein and it's passive, okay, meaning it requires no energy, right? What would we call it if we're using a channel or a protein that is passive?
Facilitated transport.
Facilitated transport, exactly. Now, if it was active, then active transport means that it requires energy, right? And we're going against our gradient. What would be a good example of active transport? What was one that we knew about?
Sodium potassium pump.
Yes, the sodium potassium pump is your number 1 you want to use for that. Okay, so one way is through the phospholipid bilayer, and these are all the things we need to think about then. Okay, then a second way is using a channel or a carrier, and if it's a protein, it could be larger and it could also be what? Charged. That's why one reason we might use a protein. Okay, then the third way to do it would be what? What's the third try? Yes. Yes. Okay. So your third way is using the whole membrane.
Oh, but I've taken the same.
Hi guys.
Oh.
To use the whole membrane.
I don't.
And if we're talking about the whole membrane, we've got 2 big categories. What are our 2 big categories? Endo and what? Exo. Exo is when it's going out. Either way, both of these, if we're using a whole membrane to go in and out, what kind of transport is that? Active, good. Which means what? Yeah, it requires some sort of energy to do it. Now we had 3 types of endocytosis bringing in. Think in your head first, what are those 3 types? Okay, now tell them, yeah, tell them to me.
Phagocytosis.
Pino, phago, and receptor mediated. Okay, so pinocytosis is cell drinking, phagocytosis is cell eating, and receptor mediated is when they bound to the proteins, right, and the receptors, and then they went in. Okay, but either way, this would be active transport. Using a protein, if you go against the gradient, would be active transport. If you're going with the gradient, it would be passive, and diffusion is always passive. And there's one other thing I would throw on there. When you're dealing with water, you could also be using a protein if you're using an aquaporin, right? Okay. ask the person you're sitting next to, do you have any issues with, I know this is not a good picture, but would you like me to Instagram this picture? Would this be helpful to you or no? She's like, no. Oh, you don't have Instagram. Like, I don't want it. Okay, so now let's go back to our question and see what it said. It says, describe the model of a cell membrane of a eukaryotic cell. Now, the model that they're referring to is called the fluid mosaic model. Okay, fluid mosaic. So that's because those phospholipids, okay, have a fluid. It's a phospholipid. It's like oil, you know, that you have. The mosaic part of your model are the proteins that are embedded in there. And we know those proteins can move around because I talked to you about a cell where they hybridize it and like a mouse protein with a person cell when they put the animal cell and a mouse cell together, the cell membrane, and they could see that the proteins that were just mouse and the proteins that were just humans, that you could see they had intermixed throughout there. So we know the proteins are moving around. And we know in our fluid mosaic model that it's the main component is the phospholipid. You have peripheral and integral proteins, you have your cholesterol, and you have your carbohydrate side chains on the outside, and your called your glycocalyx, and then your cytoskeleton to the inside. So that would be describing the model, and then discuss the different ways substances cross the membrane. That's when you would talk about, you know, diffusion through the phospholipid, or using a protein, or using a carrier. All right, are we good on that one? Okay, so now let's look at the next question. A laboratory assistant prepared solutions, 0.7, 0.5, 0.3, 0.1 molar sucrose, but forgot to label them. After realizing the error, he took, it's supposed to say he took off to Mexico, and then his assistant randomly labeled the flask again, making these four unknown solutions. He just labeled them A, B, C, and D. What experiment could you design based on the principles of diffusion and osmosis that you could use to figure out those unknown solutions? Now, did you do one of your pre-labs in here? Could you have figured out, if I had not labeled those solutions carefully, could you have figured out, deduced, or inferred from your data which solution was what from your graph as I'm looking at your, your data? Could you have figured it out? Okay. What would you have said? Well, what kind of experiment could you have set up in order to figure that out? If I was going to do a flow chart, what would you say I should do?
Get dialysis tubing.
Get dialysis tubing, absolutely. What else are we going to need?
Distilled water.
Yeah, we're going to need a beaker, right? We're going to need some distilled water out here. Sorry, that just means double distilled water. And I would say that that double distilled water is definitely going to be what? Yeah, hypotonic. And we're going to know that the water potential of that is 0. Okay, then you said what? Dialysis tubing, right? So I'm going to have some dialysis tubing in here. Tie it with some sort of string here. And what am I going to put inside that dialysis tubing? Yeah, I'm going to put the unknown solution, which in this case, and I would repeat it, 1 tube, 1 for solution A, do it again for B, C, D, etc. Yeah? And what would our hypothesis be about this?
The greater the molarity of the solution inside, the more massive.
The greater the molarity of your unknown, keep going.
The more massive, the more it'll gain in mass.
The more it would gain in mass. So then that's the big premise there. Because any molarity you have, the water potential, no matter what, is going to be what kind of number? Negative number. The more negative it is, the reason why it would have be more negative for its water potential is because it has more solutes mixed into that solution, right? And so I'm assuming I could do all of, what would be a control I could do for this experiment? What would be a good control for this experiment?
Distilled water.
Distilled water. right? Because that shouldn't change any mass. So I throw in my distilled water, then all my unknowns, whichever has the highest mass. Obviously I have to leave room for expansion, wet the strings, all those kind of things that you did in your lab. Whichever one had the greatest amount of mass, percent change in mass, I'm going to assume that that is the, what one? 0.7, right? And then working my way down in mass change. Right? So then let's look at what it says here. A description of how you would set up and perform the experiment. So I would need to explain that process of how I would set up that experiment. Okay, and think about flow charts and what you do for me, the general premises of that experiment. B, the results I would expect from that experiment. So we talked about that. I would assume whichever one had the highest molarity would have the lowest water potential and have the greatest percent change in mass. And then an explanation of those results based on the principles involved. I would need to explain about diffusion and osmosis, right? I would need to explain that, and then I would need to define what diffusion is and define what osmosis is. I would need to explain what water potential means so that I could use that to support my claim that I'm making. All right, anybody have any questions about that one that you want me to go over? No?
On the AP test, can you draw diagrams?
You can, but they have to be supportive to whatever you're saying. Any diagram things you'd actually be drawing are more like finish this graph. Or what do you think is going to happen next? Those kind of things. Yeah, predictions based on what you understand. Okay, let's look at the next one. Enzymes are biological catalysts. What do catalysts do in general? What do catalysts do?
Start a reaction.
Yeah, they get a reaction, right, to go faster. So the first one is, the first part here, it says, relate the chemical structure of an enzyme to its specificity and catalytic ability. So the first thing I would want to talk about is, what is the structure of an enzyme? And they are primarily built out of what? Proteins. So then I would have to say this protein has a primary, secondary, tertiary, and possibly a quaternary structure. So the primary structure is the sequence of what? Amino acids. The secondary is going to be the same. What are our two choices for secondary? Alpha helix and beta pleated sheet. Tertiary structure is the what? Oh, due to the R groups, but it's the what? Folding pattern. Okay? And then quaternary is if it has more than one chain. So we know all proteins are specific. We've talked about it again and again because we know the shape of proteins. And shape is critical to protein or to enzyme function because it's the actual physical shape of that protein that determines the active site. And if the active site is not the right shape, it cannot bind to the right substrate. Okay? So if the substrate, when we talk about chemical structure, we know it's specific because we could describe the chemical structure of a protein and say its specificity, because enzymes are catalysts, they have to bind to a substrate. If the shape changes, because they bind to the substrate, they can no longer bind to the substrate. If they can't bind to the substrate, they're not going to have that catalytic ability, that ability to have that reaction occur. Okay, then the next part says design a quantitative experiment with data to investigate the influence of either pH or temperature on the activity of an enzyme. Now, have you done an experiment? Yes. Don't make up one that's different. Use the experiment that we talked about. So our experiment is we were using what to do what? You just got through doing it over a course of a couple days.
Catalase to break down hydrogen peroxide.
Okay, so you were taking this experiment, H2O2, right? And you were using the enzyme catalase to break it down into what? To water and oxygen. Okay, that was the premise of your experiment. So if we look at the instructions here, it says design and to investigate the influence of either pH or temperature on the activity of the enzyme. So in our experiment, right, we took our apparatus and we added in H2O2, and we would say that this was our substrate. And we added catalase, which was our enzyme, okay, into this. And then we said it would generate what? O2. And we used a device, a probeware, okay, to basically measure the amount of O2 we had. We did it in parts per trillion per minute, the rate of oxygen production. So based on what they said here, the influence of pH or temperature, how could we alter this experiment to measure? We changed the concentration of the hydrogen peroxide and looked at the production of O2. How could we change this experiment to either incorporate temperature or pH as our independent variable?
Heat up
Heat up the catalase, right? So we could take the catalase and we could alter the temperature of the catalase. We used room temperature catalase. We could repeat the same experiment, keep it 3% hydrogen peroxide, full strength hydrogen peroxide, but instead have catalase at 10 degrees Celsius, at 32 degrees Celsius, at 64 degrees Celsius, and see what was the parts per trillion of oxygen we generated. Okay? Along that same basis, what else could we do? What?
Change the pH.
Yeah, you could change the pH of catalase. We could have catalase mixed in with, just as itself, at the substance it comes, or we could mix our catalase with some vinegar or some sort of acid, and with some baking soda and water, some sort of base, and see if catalase still created the same amount of product, whether it was mixed in, had water added in with it, or had an acid mixed into it, or we added a base mixed in with it, and see if it made the same rate of oxygen production. And that's how we could alter our experiment to include a change in pH or temperature. And then the third part says, describe what information concerning the structure of an enzyme could be inferred from our experiment. Well, we know that enzymes have an optimal what? pH and an optimal temperature. And that if we change the pH, we could change the allosteric, the shape of that enzyme. If we change the shape, what are we going to change again? The active site, right? Change the shape, you'll change the active site. If we change the pH, we can change the active site. And so then we would affect its catalytic ability. All right. Now, the next set of questions that we have in that document are about photosynthesis and cellular respiration for our essay prep. So that'll be in the part 2 essay prep, not in part 1. And then the non-PDF part, we had another one as well. Let me get in here. Oh, hello. Okay, then. Oh, here it is, resources for review. Oh, my bad. Here we go. So then we had the grid-ins here as another source of potential questions. So we could talk about in this one, number 4, right? Potato cores were placed in solutions of varying concentrations were found to neither gain nor lose mass in a sucrose solution of 0.32. That sounds exactly like our lab, right? So use this information to calculate the solute potential of the potato cores. The temperature is 22 degrees. So we would have to get the temperature, we'd have to do the 20, to get it to Kelvin, right? Don't you have to add it to 273, right, to get a Kelvin? And then we've got our 0.0831 liters per bar per moles per Kelvin. And then we have our C number. Our C number would be what? 0.32. And what are we in? sucrose, our ionization constant is 1 and then negative, and then you would just calculate that out. And I would do that and see if you agree with your friends on the number that you come to. Okay, and then this one is dealing with cellular respiration, so we haven't gone over that yet, or that. Okay, but we could look at 6. An enzyme in the liver removes a phosphate group from glucose. Oh, wait, maybe we need to wait on that one. I think, wait, let me look at it. Oh, this is just a rate. This is exactly like what we did. All you have to do is calculate a rate, right? Final minus initial over time. So you could do that one as well. Okay, and then we'll hit the other ones because I know you need to go to class. But don't forget to look at those as well. Are you glad you came today? Understand a little better, my quiet children? Okay. They do not talk, but I love them so much. Have a good day. Hope you're studying. Okay. Love you much. Bye.